All right, as we continue here, I want to remind you that we're looking at organisms that are in the kingdom Protista. And within the kingdom Protista, there are plant-like protists that we call algae, and there are fungus-like protists that we call slime molds and water molds, and there are the animal-like protists, which are what we're starting with here. And so these phyla that we've been going through are animal-like protists known as Protozoa. Now, the term Protozoa is not the kingdom name, so don't confuse Protozoa with Protista. Protista is the kingdom. Protozoa is just a common name that we use to refer to the animal-like protists. So we've already looked at the phylum Ciliophora and the phylum Zoomastigena. So we're going to take a look at the Phylum Sarcodina, and then the remaining one is Phylum Sporozoa. And the Sarcodines, and remember that they're classified by how they move, their mode of locomotion, their locomotive structures, or their lack of locomotive structures, as in the Phylum Sporozoa, as we'll see on the next slide. So this is the only slide for the Phylum Sarcodina. And Sarcodines include foraminiferans, amoebas, and heliozoans, along with others, but these are some of the biggest. So this figure represents foraminiferans. And I know this looks like something that washed up on a beach, like a bunch of seashells, but these are actually microscopic. And they look like seashells. They're kind of in the shape of seashells, and they're made out of the same substance as seashells, calcium carbonate, but they're actually microscopic, and these would be shells that a very small one-celled amoeba would be living inside. So foraminiferans are basically amoeba that have a calcium carbonate shell around themselves. And then you might be asking yourself, well, what's an amoeba? Well, this diagram is an amoeba, is representing an amoeba, and these are amoeba. And one thing you might note about them is no two of them look the same, and are the same shape anyway. And that's because they're shapeshifters. They can take on any shape they want by extending pseudopodia in any direction they want. And that's the common characteristic and locomotive structure that we find in the Phylum Sarcodina. Sarcodines move using these false feet known as pseudopodia. Pseudo means false. Podia refers to feet. Or pseudopod, one of them is called a pseudopod, false foot. And it's false because it's temporary. This amoeba, for example, is extending this pseudopod in this direction, but it might, once it moves in that direction, it might change its mind and extend a pseudopod in the opposite direction, or sideways, or whichever way it wants to. So these things never look the same way twice when you look at them. They're always going to be a different shape. And then finally, as an example on this slide, we have the heliozoans, and helio refers to sun. So heliozoan, literally, and zoa always refers to zoo or animal. So this literally translates to sun animals, and you can see why they call them sun animals. They look like a starburst or something radiating like the sun radiates. And you should recognize this as being an example of this kind of sarcodine is what you're looking at in the lab that was known as actinospherium. So this looks like actinospherium. And these rays that are coming off of the heliozoan are pseudopods, but they're specialized pseudopods that we refer to as actinopods, actually. So amoebas, foraminiferans, heliozoans. Oh, and by the way, foraminiferans, we saw those back when we were studying the Earth and life through time, and how fossils are used to, index fossils are used to date other fossils. So these guys make great index fossils because they've been around for millions of years, and their species have changed over those millions of years, and they are found all over the world. So they're a great index fossil. However, they are microscopic, so they would be micro-index fossils. So as I mentioned, sarcodines have pseudopodia, false feet, that function in locomotion and feeding. So it's not only their locomotive structure, it's also how they feed, as we can see here in this inset. So when it moves, it extends its ectoplasmic gel, which is a semi-solid. And then there's a more fluid endoplasmic sol, So gel refers to a semi-solid, and sol refers to a liquid, like the cytosol. You should be familiar with that term cytosol, which means the aqueous solution or liquid portion of the cytoplasm. And when you look at one of these under the microscope, as I hope you have by now in the lab, you can see the cytoplasm streaming out into the pseudopodia. So when it puts out a pseudopod, the first thing it does is extend its plasma membrane, and just under the plasma membrane is this gel-like portion of the cytoplasm, the ectoplasmic gel, and then there's a core of liquid endoplasmic sol that will stream into the pseudopod. And you can watch that because all the components within the cytoplasm are flowing along with the sol, the liquid portion, and that's known as cytoplasmic streaming. So I have added some arrows here just to represent that, cytoplasmic streaming, and of course labeled it. You ought to add that too, just to remind you of what cytoplasmic streaming is. It's the streaming of the cytoplasm out into the pseudopod, and you can actually watch that happening in a living specimen, like you did in the lab. So it's very misunderstood how they do this, but it seems to involve enzymatic conversion between gel and sol. In other words, there are enzymes that will turn the semi-solid gel into sol, and sol back into gel. So it's the conversion between gel and sol that allows for this kind of locomotion or what we call amoeboid movement. So because it's how amoebas move, we call it amoeboid movement. But there are cells that move by amoeboid movement that are not amoeba. For example, macrophages, which are part of your innate immune system. They're crawling around inside your body, and the way they're crawling around is by amoeboid movement. And there are white blood cells that do the same thing. So not only amoeba use amoeboid movement. And that also exemplifies a possible evolutionary link or common ancestry between animals like us and sarcodines like amoeba, or animal like protists in general. So they feed by phagocytosis. And if you'll recall, that is a form of bulk transport, bulk endocytosis, right? Endocytosis is a general term, and then there are more specific ways that cells will bring large quantities of things inside the cell, one of them being phagocytosis, which means cell eating. Phago means to feed, and cyto refers to cell. So it's the process, the cis is kind of the process, so it's the process of cell eating. They extend their pseudopodia, and we're seeing that happen here. There are two pseudopodia that are being extended around a food item here. And then the pseudopodia, the plasma membrane of the pseudopodia, will fuse together and form a food vacuole around that food item, and it will then be inside the cell. The food vacuole will be inside the cell, just like the food vacuoles that we see here in the diagram. So that's where these food vacuoles came from. They came from the process of phagocytosis, and they will contain food. And then lysosomes will fuse with those food vacuoles and digest the contents, and the nutrients will diffuse out into the cytoplasm. And then finally, as all protozoa, sarcodines reproduce asexually by mitotic binary fission. They split in two. But they are eukaryotic cells, therefore they have to divide their nucleus first, and that's mitosis, by the process of mitosis, and then the cell actually splits, that's cytokinesis. So mitosis and cytokinesis is how eukaryotic cells undergo cell division, but if cell division is for reproduction, we call that binary fission. And then finally, I don't want you to believe that no sarcodines cause disease, because when we were looking at the phylum Zoomastigana, all I showed you were diseases that were caused by zoomastigonins, even though most zoomastigonins are not pathogenic. And most sarcodines are not pathogenic either, but some of them do cause disease. You may have heard of an amoeba that can get in your eye when you swim in water that can cause you to go blind. There is also this problem, amoebic dysentery. This is a fecal smear, so in other words, this is poop smeared on a slide, and this person is infected with Entamoeba histolytica, which causes amoebic dysentery, which is severe diarrhea. But these big clear things in this fecal smear are the amoeba. So usually poop doesn't look like this, with all these big clear round cells within the feces. But this shows that this person has been infected with amoebic dysentery and they have severe diarrhea. And diarrhea can be life-threatening because it dehydrates you primarily. That brings us to the phylum Sporozoa, and sporozoans are all pathogens. So it's not like the case in Zoomestigina or Ciliophora or Sarcodina, where most of them are non-pathogenic. These guys are all pathogens. They're all parasites. Well, let me back up. They're all parasites, and some of them, because they're parasites or pathogens, they cause disease. But they're non-motile, and so they don't have cilia, they don't have flagella, and they don't form pseudopodia. They are carried from person to person or organism to organism, either by eating, or passed on from organism to organism by eating, or by injection. So on the next slide we'll be looking at malaria, and that's an example of injection, where mosquitoes take the organism from one person and inject it into another person. And then once they're in the organism, they're carried around by bodily fluids. They're transported around in the body by bodily fluids like blood. So the disease we're looking at here is toxoplasmosis, which is caused by the organism Toxoplasma gondii. And you'll notice that there's a pregnant woman here, and you may have heard that pregnant women should not, if the household has a cat as a pet, the pregnant woman should not be cleaning out the litter box. And that's because this disease can be passed on to a pregnant woman by the feces of the cat, because of these oocysts that are in the feces of the cat. And the danger is that not so much that the woman, the mother, will be contracting toxoplasmosis, but that can be passed on to the baby. That's known as congenital toxoplasmosis, where the mother passes on the disease and the organism to the baby. That's a congenital contraction, or not. That's a congenital way to contract the disease. In this life cycle, we see that the primary host, which we also call the definitive host, is the cat, because that's where Toxoplasma sexually reproduces. So we see here in the diagram, sexual reproduction is taking place in the cat, and that's true for any parasite or pathogenic organism. The species in which it reproduces sexually is considered the primary host or the definitive host. They mean the same thing. The other hosts that we see here in the diagram are all considered secondary hosts. And Toxoplasma gets into its primary host, in other words the cat, when the cat ingests an infected mouse. So this is the primary life cycle of the parasite. But it can break out of this life cycle and infect other organisms. And again, those would be, well, the mouse is the secondary host, but other organisms can be the secondary host too. Generally, other mammals that would somehow contract the sporozoites that have come out in the feces of the cat. And that can be in soil, or it can be on the hands of a person who cleans out a litter box. The sporozoites, when they're ingested, will cause the disease. But also these, the other form of the organism called a bradyzoite will insist in the tissues of the animal that's infected. And when someone or something, some other animal eats the tissues, they can contract the organism. Just like when the cat eats the infected mouse. And finally, as I mentioned, congenital transmission is transmission from mother to fetus, which can happen in all hosts. That can happen in the cat, in the mouse, in cattle, in pigs, and in humans. So that's something to be concerned about with this disease. The sporozoan disease that we are most concerned about, really, is malaria, because it infects and kills so many people in the world. So Plasmodium falciparum is one of the organisms that causes malaria, but there are others. But notice they all are Plasmodium, which is the genus name of the scientific name, and then the second name of the scientific name is the species name. So these are different species, but they're all in the same genus, Plasmodium. Over 1 million people every year are infected, and mostly children under 5 years old. And as you can see here in the diagram, it is passed on by the vector, the Anopheles mosquito. So the vector and primary host is the Anopheles mosquito. Remember, primary host or definitive host means that that's where Plasmodium sexually reproduces, is within the Anopheles mosquito. So the rest of what is coming up here is a description of what's going on here in the life cycle diagram. I'm just going to describe it on the diagram, and it would probably be better for you, instead of just copying everything that is written here, to just go ahead and label it on the diagram. So it starts when a person is bitten by an Anopheles mosquito that injects, remember that sporozoans can't move on their own, so it injects the sporozoites, as we call them, which are in the saliva of the mosquito, into the person. The sporozoites then find their way through the bloodstream. They travel through the bloodstream. They don't swim, and they're carried to the liver, where they asexually reproduce and produce these things called merozoites. So they actually undergo a transformation from one form, sporozoite, to another form, merozoite, which are specialized to travel in the blood and infect red blood cells. So when they come in contact with red blood cells, they actually can enter the red blood cells, and then they asexually reproduce more and produce more merozoites, but they will also produce gametes. And then the cells are destroyed, they break open, they release the merozoites, which will go on and infect other red blood cells. So you can see how this can spread very quickly and destroy red blood cells and cause anemia. Anemia is a condition where you don't have enough red blood cells, or you don't have enough hemoglobin to carry oxygen in your blood. So you can't live without being able to transport oxygen, and that's what kills people that are infected with malaria. But again, some of these are gametes. So the gametes are circulating around in the person's blood that's infected with malaria, and an Anopheles mosquito will come along and bite that person and pick up those gametes. And fertilization then takes place, producing zygotes, and that's the only diploid of this organism in this life cycle. In other words, when sperm, which have half the number of chromosomes, fertilize an egg, which have half the number of chromosomes, remember half is haploid, that produces a zygote, which is one cell, but it has the full number of chromosomes. So that's diploid. Then it immediately undergoes meiosis, which reduces the number of chromosomes to half again. So that means that everything else in this diagram, the sporozoites and merozoites, are haploid, and the gametes are haploid. The only diploid in the whole diagram are the zygotes that result from fertilization. And those then develop into sporozoites, which are injected into another person, if that mosquito bites another person, which is highly likely. So that's the problem with malaria, and one form of control of malaria is trying to control the mosquitoes that pass them on. So there's a lot of work going on in that area, along with vaccines. But we haven't been able to develop a 100% effective vaccine for malaria. And finally, we're taking a look at the ecological impact of the protozoa. And we also get into some other protists here that are involved. But the first one is as a mutualistic symbiotic relationship with other organisms. For example, I'm sure you know that termites eat wood, but the termites themselves cannot actually digest the wood. Their digestive system can't digest the wood. It's actually the protozoa, the zooflagellates, or though the members of the phylum Zoomastigina, if you'll recall, they locomote with flagella, and if you look closely here, you can see that these organisms are highly flagellated. So they are zoomastiginid protozoans that live inside the gut of the termite. If you break open the gut of a termite and look at the contents under the microscope, you'll see that they are packed with these zoomastiginid flagellated protozoans. And they are the ones that are actually helping to digest the wood. And that's what we're seeing here inside these protozoans. This is wood inside the protozoan. So this is one protozoan, and that's the wood in the food vacuoles within that protozoan. But actually, there are bacteria that live within the protozoa that create the enzymes to actually be able to break down the constituents of wood, which is lignin and cellulose. So it's almost like a Russian doll situation where the termite has protozoans living inside that help digest the wood, but the protozoans have bacteria living inside them that digest the wood. So it's endosymbiosis within endosymbiosis going on to allow termites to be able to digest wood. Phytoplankton are not protozoans, they are algae. Remember, there are plant-like protists that are known as algae, but I'm bringing them up here so that you can understand the ecological role of the zooplankton, which are partially made up of protozoa, the animal-like protists. So algae are the producers in aquatic ecosystems, whether it be a marine ecosystem like we're looking at here, which means saltwater, or a freshwater ecosystem. Algae are major primary producers. They're photoautotrophic primary producers. Then protozoa, along with animals, multicellular animals, make up zooplankton, which are primary consumers within the aquatic food web, and they serve as food. So the algae serve as food, and zooplankton, which are largely made up of protozoa, also serve as food within, at the very base of the aquatic food web. So they're a very important constituent or component of the aquatic food webs.